Alright, so now let's move on to question number 16. It is from May-June 2017, paper 1 to question 10. It says that the effect of the substrate concentration on an enzyme catalyzed reaction was measured in three different conditions. Number one condition over here was with no inhibitor. The second condition was with a competitive inhibitor, right, and with a non-competitive inhibitor. Then it says that the graph shows the result. This is the result. They have shown that this line over here is representing the graph with no inhibitor, right? Enzyme with no inhibitor. It is a graph of the enzyme with no inhibitor, right? And this is the inhibitor X. Now we have to identify that whether it is a competitive inhibitor or the competitive inhibitor, right? And similarly we have to identify the same for inhibitor Y. Then they say over here that which statement is correct. Alright, so we did a question similar to this above as well, where we talked about the competitive and the non-competitive inhibitor. Over here you guys can see that if I talk about the non-competitive inhibitor and the competitive inhibitor, so these were the things we studied about the competitive and non-competitive inhibitor. The key points basically we studied about them, right? Now for the non-competitive inhibitor, we have studied that for the non-competitive inhibitor the Vmax decreases and the Km stays the same. Vmax is the maximum rate of reaction, right? And for the competitive inhibitor the Vmax stays the same and the Km increases, right? So we can clearly identify the following, that which is the competitive and which is the non-competitive by the Vmax. This is the Vmax, which is the maximum rate of reaction, right? Vmax is staying the same for the inhibitor X. When the inhibitor X is there, the Vmax is staying the same, which means that inhibitor X is a competitive inhibitor. Vmax is decreasing with the inhibitor Y, which means that inhibitor Y is a non-competitive inhibitor. Hence the correct answer over here would be that Y is a non-competitive inhibitor. Yes, exactly. It is true that Y is a non-competitive inhibitor. We know that Y is a non-competitive inhibitor, so C is also incorrect, right? X is a competitive inhibitor. Alright, so and X is a non-competitive inhibitor. No, X is not a competitive inhibitor, right? Now we have to select our answer from option A and option D. Let's give option A first a read. Option A says that X is a competitive inhibitor which binds to the other side than the active side of the enzyme. No. Competitive inhibitor is similar in shape to the substrate, and it binds to the active side of the enzyme, right? It does not bind to the allosteric side other than the active side of the enzyme is the allosteric side, and the non-competitive inhibitors basically bind with the allosteric side, right, not the competitive inhibitor. Hence option A is also incorrect. Now option D says that Y is a non-competitive inhibitor which binds to the side other than the active side of the enzyme, which means that it binds to the allosteric side of the enzyme, which is a site other than the active site of the enzyme. So hence the correct answer over here for question number 16 is option D, that Y is a competitive inhibitor which binds to the site other than the active site of the enzyme, right? I hope that this MCQ is clear to all of you. Alright, so now let's move on to question 17, which is from May-June 2017, paper 1-3, question 10. It says that the graph shows the effect of an enzyme on a reaction. Which labels are correct? Alright, so basically let's see that what these labels are basically telling us. Now from the substrate to the peak, we have the activation energy, right? It is the activation energy. Now there are two graphs shown, so the activation energy for the Y graph is less and the activation energy for the X graph is more, right? And the W is basically representing the change in the activation energy. From the substrate to the product, we have the delta H, which is the enthalpy change. Now let's read the statements one by one and try to get the correct answer. X is a reaction without the enzyme. X is a reaction without the enzyme. Yes, this is true that X is a reaction x हमारे पास over here, what is x over here? X is being represented by these lines, right? And these lines, it is x is a reaction without the enzyme. Why is it is the reaction without the enzyme? Because the activation energy for this reaction is high. We know that enzymes are the biological catalyst, right? Which means that they speed up the chemical reaction by lowering the activation energy, right? And over here in the x, the reaction, in the x reaction, the activation energy is high, right? And w is not the activation energy, it is rather the change in the activation energy, right? It is the change in the activation energy, so hence a is incorrect, right? Then let's move on to option D. Option B, it says that x is a reaction with the enzyme. No, x is not a reaction with the enzyme because it has a very high activation energy, right? Y is a reaction with the enzyme because it has a low activation energy, so hence this is also incorrect. Then it says that y is a reaction with the enzyme. Yes, y is a reaction with the enzyme. How we can conclude it? With the low activation energy, right? Because enzymes, we know that enzymes lower the activation energy. W is the difference in the activation energy. Yes, already I told you that W is basically the difference in the activation energy. Hence, the correct answer for this MCQ 17 is option C over here. I hope that this MCQ is clear to all of you. Alright, so now let's move on to question 18, which is from October-November 17, paper 1-1, question 10. It says that lysozyme is an enzyme found in the tears and the saliva. Which statement refers only to the tertiary structure of the lysozymes? Lysozymes, they are talking about that they are the enzymes in nature, right? And they are saying that which statement is referring to the tertiary structure of the lysozyme, right? The 3D structure of the enzyme. A section of molecule consists of an alpha helix. Alright, so if I talk about the alpha helix and the beta helix structure, so it represents the secondary structure of the enzyme, right? It consists of a single polypeptide chain. We know that in the quaternary structure there are more than one polypeptide chain, right? So it may be representing the primary, secondary or tertiary structure, but it is not representing the quaternary structure obviously. But let's look at this option again once we read all the other options, right? It is 129 amino acids long, right? It is also representing the primary structure of the protein, right? Because the primary structure of the protein basically tells us about the sequence of the amino acid in a polypeptide chain, right? The 35th and the 52nd amino acids help form the active site. Alright, so we know that enzymes are globular in nature, right? And due to the folding of the enzymes, due to the folding of those polypeptide chain into the 3D structure, basically the active site is formed, right? So hence option D over here is the correct answer, that the 35th and the 52nd amino acid helps to form the active site. Alright, so question 19 over here is from May-June 2017, paper 1 to question 12. It says that how is the Mitchell's-Menten constant Km used? We know that what is basically Km, it is the substrate concentration at half Vmax, right? And we know that if the Km is high, the affinity between the enzymes and the substrate is low, right? And vice versa, that if the Km is low, then the affinity between the enzymes and the substrate is high, right? Now they are saying that how is the Mitchell's-Menten constant used? So let's look at the options one by one. To access the efficiency of an enzyme in catalyzing a reaction. Okay, let's read the other options as well before concluding the correct answer. To compare the affinity of the enzyme to their substrate. To find the maximum velocity of the enzyme. No. To find the rate at which the substrate is loaded by an enzyme. No. The best possible answer over here is to compare the affinity of an enzyme to their substrate. As we know that if the value of the Km is high, so the affinity is low, and if the Km value is low, so the affinity is high between the enzymes and the substrate obviously. So hence the correct answer over here would be option B. Let's now move on to question 20. Question 20 over here is from May June 17, paper 1-3, question 10. It says that the graph shows the energy changes in a chemical reaction. Alright. What is the activation energy? What is the activation energy when the enzyme is added? We know that enzymes work by lowering the activation energy. Lowering the activation energy. Enzymes work by lowering the activation energy. So we can clearly see that the lowest activation energy is seen by this graph particularly, right? This is the activation energy for this graph and this is the activation energy for the up graph, okay? For the graph which is upward. So hence the graph where the enzyme is added is this one, right? This is where the enzyme is added. So they are saying what is the activation energy when the enzyme is added. So this is the activation energy where enzyme is added, which is representing by 2 only. Hence the correct answer over here would be option B, 2 only. Right? They are talking about the activation energy. So activation energy for the reaction where the enzyme was added is represented by 2. I hope that this MCQ is clear. Now let's move on to question 21. Alright, so now let's move on with question 21. It is from May June 2017, paper 1-3, question 11. It says that the graph shows the results of an investigation into the effect of amylase on the hydrolysis of the starch at 3 different temperatures. In each investigation, the concentration and the volume of solution was kept constant. What conclusions may be drawn? Alright, so they are basically investigating the effect of the enzyme amylase on the hydrolysis of starch on 3 different temperatures, 50, 60 and 70. We know that amylase hydrolyzes the starch and the product of the hydrolysis is basically the reducing sugars, right? So more the reducing sugars are present, right? More the reducing sugars are present, it means that more good hydrolysis or more faster the hydrolysis is being taken place, right? Now let's look at the options one by one and the graph obviously, and let's conclude that what conclusion may be drawn, right? 50 degrees Celsius is the optimum temperature of the amylase. Alright, we can say that it is the optimum temperature of the amylase. Why? Because we can over here see that the concentration of the reducing sugar is hitting its maximum at only 50 degrees Celsius. But we never know that maybe at the 40 degrees Celsius it may hit even more further, right? Even the concentration of the reducing sugar may be even more than what is over there at the 50 degrees Celsius. This can also be the case, right? So we cannot comment on the optimum temperature of the enzyme amylase, right? This conclusion cannot be drawn from the graph. So hence statement A is incorrect. Now let's look at option B. It says that at 60 degrees Celsius all the starch is hydrolyzed after 30 minutes. Alright, so they are talking about the 60 degrees Celsius and they are saying that all the starch is hydrolyzed after the 30 minutes. So no, this is not correct. Maybe what may happen over here that we cannot explain it that at the 60 degrees Celsius all the starch is hydrolyzed after 30 minutes. No, not after 30 minutes. After 30 minutes you guys can see that the concentration of the reducing sugar is constant, right? So we cannot say that all the starch is hydrolyzed after 30 minutes, right? Because the concentration of the reducing sugar is not increasing, it is staying the same. So hence this statement is also incorrect over here. Then the rate of the hydrolysis of the starch is faster at 50 degrees Celsius than at 70 degrees Celsius. Alright, so let's see over here if we talk about this point and if we talk about the concentration of the reducing sugar at 50 degrees Celsius, and then if we talk about the concentration of the reducing sugar at this point for the 70 degrees Celsius. So the concentration of the reducing sugar at 70 degrees Celsius is more, right? Which means that the hydrolysis is faster in the 70 degrees Celsius rather than the 50 degrees Celsius, right? So it would be 70 degrees Celsius. The hydrolysis of starch is faster at 70 degrees Celsius than at the 50 degrees Celsius, right? So this statement is also incorrect. Then if we look at option C, it says that at 70 degrees Celsius the amylase is denatured before the hydrolysis is completed, right? At 70 degrees Celsius, you guys can see over here. Let me clear the graph so that we can easily interpret. Now they are saying that at 70 degrees Celsius, the amylase is denatured before the hydrolysis is completed. Yes, obviously it is true. Why it is true? Because you guys can compare it with the other graph, that in the other graphs the concentration of the reducing sugar is reaching more heights than at the 70 degrees Celsius, right? So it is denatured before the hydrolysis is completed. Hence the concentration of the reducing sugar is the least over here. So hence the correct answer over here would be option C. I hope that this MCQ is clear to all of you. Let's now move on to question 22. Question 22 over here says that question 22 is from October-November 15, paper 1-3, question 12. It says that the graph shows the concentration of one of the substance which is involved in the enzyme catalyzed reaction. Which substance is shown by the line X? Alright, so we know that the concentration of the substrate changes with the time. Why? Because substrate is going to break down to convert into the product, right? So hence the concentration of the substrate would not remain constant throughout the reaction, right? Similarly, as the substrate finishes, so the enzyme substrate complexes also stops forming, right? So hence C is also incorrect. Then if I talk about the enzyme product complex, if the substrate is finished, right, so no substrate is there to break down into product. I hope that you are getting it what I am trying to say, that if the substrate is finished, so no substrate is going to convert into the product. So hence the enzyme substrate complexes will be there at one point and then will not be there at the other point. Similarly, if I talk about the enzyme, so in enzymes, what we can say over here, that the concentration of the enzymes will remain constant. They will remain unchanged at the end of the reaction as well, right? They are aiding the metabolic reactions in the body, but they are remaining unchanged at the end of the reactions, right? So hence the correct answer over here would be option A. Alright, so now let's move on to question 23 over here. It is from May-June 2014, paper 1-1, question 12. It over here says that the statements are about the enzymes. They have then given us a list of 4 statements about the enzymes, and then they say that which statements are correct for all enzymes. Now they have highlighted the word all over here, that which statement is correct for all of the enzymes. So we know that enzymes are globular proteins. Yes, this is correct. It is a statement from our syllabus as well. All enzymes are tertiary in structure, right? So enzymes are the globular proteins, so hence this statement is absolutely correct. So 1, yes, it is correct. Let's eliminate B over here, because B is not having 1, obviously that's why we have eliminated B over here. Then they say that they are formed in the smooth endoplasmic reticulum. We know that enzymes are protein in nature, and proteins are synthesized in the ribosomes, and ribosomes are attached to the rough endoplasmic reticulum, right? Rough endoplasmic reticulum, not the smooth endoplasmic reticulum. So hence it is incorrect. They are only found attached to the plasma membrane in the cell. No, they can be found freely in the cytoplasm as well. They are the enzymes are of 2 types, intracellular enzyme and the extracellular enzymes. We have both type of enzymes, right? So hence statement 3 is also incorrect. Now statement 2 and 3 are incorrect, so let's eliminate C over here as well, and let's eliminate D over here as well. So I think we have an obvious answer, which is A over here, 1 and 4 only. Now the fourth statement is a little contradictory, that they can be inhibited by competitive inhibitors, and then in the question they are saying that for all enzymes, some of the enzymes can be inhibited by non-competitive inhibitors as well, right? Non-competitive inhibitors as well. Not all enzymes can be inhibited by the competitive inhibitors, rather some enzymes can be inhibited by non-competitive inhibitors as well. But if we look at the structure of the question, and if we look at the options which they have given us, so A is the best possible answer over here, right? Otherwise the competitive inhibitors as well as non-competitive inhibitors both can inhibit the activity of the enzyme, right? But for this question, as per the options given over here, option A is the best possible answer, right? So let's move on to question 24 now. Alright, so now let's move on to question 24 over here, which is from October-November 2015, paper 1-3, question 13. It says that what describes the induced fate mode of the action of an enzyme. Right, to solve this question, you must have a prior knowledge of the induced fate theory from the enzyme chapter. What does the induced fate theory basically states that the enzyme's active side, the active side of the enzyme, if I talk about the active side of the enzyme, basically they say that the active side of the enzyme is not a perfect fit for the substrate initially, right? Instead, the enzyme undergoes a conformational change when the substrate binds to it, improving the basically fit of the substrate to the active side of the enzyme, right? So hence the correct answer over here would be option C, right? The substrate causes a change in the enzyme shape so that the active site can bind, right? This is the key feature of the induced fate theory, making it the correct answer over here. If I talk about the other statements as well, the binding of the active site to the substrate causes the enzyme to change the shape. This applies to the lock and key and the induced fate theories. It applies to the both, right? The lock and key as well as the induced fate. So it is not unique to the induced fate model, right? And then if I talk about the option B over here, it says that the substrate and the active site has the complementary shapes that form the temporary bond. This statement is more aligned with the lock and key hypothesis, right, where the substrate is already a perfect fit. So this is also not correct, as it is for the lock and key hypothesis. Then if I talk about option D, it says that the substrate changes the shape so it can bind to the active site. This feature that the substrate changes the shape, so the substrate does not changes the shape, right? Basically enzyme changes the shape of the active site so that the substrate can easily bind to it, right? So hence the correct answer for this is option C. Alright, so now let's move on to question 25. It is from October-November 13, paper 1 to question 11. They are saying that what is correct for the non-competitive inhibitor of the enzyme action. Then they have given us the set of the three statements, and we have to select the correct statement for the non-competitive inhibitor of the enzyme. We know that the non-competitive inhibitor of the enzyme binds to a site other than the active side of the enzyme. For example, this is the active side of the enzyme and this is the allosteric side of the enzyme. So the side other than the active side of the enzyme is the allosteric side to which the non-competitive inhibitor binds, right? Non-competitive inhibitor does not bind to the active side. Now they are saying that increasing the concentration of the enzyme substrate will reduce its effect. It is a key feature of the competitive inhibition, right? Competitive inhibition, where the competitive inhibitors are competing with the substrate, and if we increase the concentration of the substrate, the effect of the competitive inhibitors can be reduced and the enzyme activity may enhance, right? It reduces the activation energy required for a reaction to take place. We know that inhibitors basically inhibit the enzyme action. They inhibit the enzyme action. So they reduce the activity of the enzyme. If the activation energy decreases, so the rate of the enzyme action may increase, right? So hence this is also incorrect. It reduces the maximum rate of the reaction. Yes, this is correct. Why? Because we know that inhibitors work by inhibiting the rate of the reaction, right? Which means that it is reducing the maximum rate of the reaction. So hence option B is the correct answer over here, 3 only. Alright, so now let's move on to question number 26, which is from October-November 2012, paper 1-1, question 14. It says that what is the effect of an enzyme in an enzyme catalyzed reaction. We know that enzyme speeds up the rate of reaction or speeds up the rate of the metabolic reactions in our body by lowering the activation energy, right? By decreasing the activation energy, but it has no effect on the overall energy change of the reaction, right? So it has no effect on the energy yield. The overall energy yield will remain the same, but however it will increase the speed of reaction by decreasing the activation energy. Hence the correct answer to question 26 is option B over here. Now let's move on to question number 27 over here. Question 27 is now from October November 13, paper 1 to question 12. It says that the enzyme lysozyme secreted from the tear glands forms the deposit on the contact lenses. Which ingredient would be effective in a contact lens cleaner for removing these deposits? Alright, so over here we know that there is an enzyme which is lysozyme, they are saying, and they are saying that they are secreted from the tear glands and forms the deposits on the contact lenses, right? And as they form the deposits on the contact lenses, so they may cause a disruptance in the vision, right? So they are saying that which ingredient would be effective in a contact lens screener for removing these deposits. So these deposits, which are the lysozyme enzymes, are basically protein in nature, so they must be removed, right? They are saying that they must be removed. So the substance which is used to remove the protein would be the proteases, right? They are the enzyme which will digest the proteins. So hence the correct answer over here would be option D, proteases. Now let's move on to the last MCQ of this whole topical MCQs of the enzyme series. Right, question 28, which is from October November 2012, paper 1 to question 13, right? They are saying that what is the role of enzymes in the metabolism. Alright, so let's read the options one by one and try to conclude the correct answer. Option A says that to catalyze the hydrolysis of large molecules only. Alright, so enzymes basically can catalyze both the catabolic reactions as well as anabolic reaction. In catabolic reactions, we know that, for example, a product C breaks down into A plus B, right? And in anabolic reaction, A plus B combines to form C, right? So enzymes are not only responsible to hydrolyze the large molecules, but however they can form the large molecules as well, right? So this option is incorrect. To increase the number of collagen between the molecules, alright, let's look at the other options as well. To lower the activation energy required to start the reaction, to supply the activation energy required to start a reaction. Basically enzymes are acting as a biological catalyst, right? They speeds up the chemical reaction by lowering the activation energy. Hence the correct answer to this question is option C, to lower the activation energy required to start a reaction, right? Alright everyone, so that was it about the topical MCQs of enzymes. I hope that you guys enjoyed solving these MCQs with me, and if you guys did, do consider subscribing this channel and giving it a like and leaving a comment that how do you find these lectures. Your appreciation means a lot to me. Till then, take care and have a nice day. See you guys in the next video.